Good afternoon, everyone, and welcome to our hybrid seminar for staff here at the LMB and for our online audience as part of Cambridge Biomedical Campus Tours. I think we've got a bit of an echo. I'll just wait 1 second. Okay, yeah. All right then. So before we begin, just some technical details. At the end of the talk, there'll be an opportunity to ask questions. If you're participating online, if you could ask those questions in the Q&A, then I will read them out so that everyone here can also hear them. So a few words about the LMB. So the MRC Laboratory of Molecular Biology, or LMB, is a world-leading research institute dedicated to understanding important biological processes at the level of atoms, molecules, cells, and organisms. We aim to study difficult long-term problems, and in doing so, to contribute knowledge needed to solve key issues in human health. Today, I'm really delighted to welcome Marta Shahbazi to give our talk. Marta is a group leader in the Division of Cell Biology. She obtained her PhD in 2013 at the Spanish National Cancer Research Centre, and in 2014, she joined the laboratory of Professor Magdalena Sinica-Goetz at the University of Cambridge to study mammalian embryo development and implantation. As a postdoctoral fellow, she developed a method to culture human embryos beyond implantation in vitro. She has used this methodology to study tissue morphogenesis, cell fate specification, and the molecular mechanisms that underlie pregnancy loss. She joined the LMB in February 2020 to understand self-organization and developmental plasticity in mouse and human embryos. I'm delighted that Marta's going to give our talk today. Thank you.
Thank you very much for inviting me. It's a great pleasure to be here. My passion for embryo development, especially human embryo development. And so before we start, I would really like to give you a historical introduction so we can discuss how scientists have approached the question of embryo development throughout history. And so we really need to start with this drawing. Has anyone seen this before? Hands up, no one? Yeah, okay, a couple of them. This is supposed to be the head of a sperm. So you can see here, the head and the tail. And inside the head of the sperm, there is a little man that needs to grow. And that little man has a sperm. And inside that sperm, there will be more little man. So we can go ad infinitum. Believe it or not, this is what people were thinking back in the 18th century. This is called the theory of preformation, which means scientists thought that embryo development entails only growth. The structures and the body are already present in the sperm, and this little human just has to grow to give rise to the baby. Of course, embryo development entails growth. We know that now. But we also know embryo development entails generation of shape, and that's what is called epigenesis. And this was the dominant theory after the 19th century. And this was thanks to the fact that with microscopes, scientists really failed to see that little man inside the sperm. So we now know embryo development entails generation of shape, as you can see there, but actually this was already proposed 2,000 years earlier by Aristotle. So okay, we know now embryo development entails growth, entails generation of form. But there is one extra thing that is really important during development, and that's generation of cellular diversity. We need to generate different cellular types. So brain cells, muscle cells, liver cells, so on and so forth. And I would like to introduce that concept with my favorite cartoon when I was a little girl. Let us see if this is going to play.
Andén número 8. Células para los músculos, andén número 10. Células para la piel, andén número 14.
Calma, niñas, hay que tener disciplina.
No os empujéis. Células para el corazón, andén número 22. Un poco de orden, por favor.
I really like this cartoon because it explains it so easily. There are these policemen telling cells what to do, where to go. We hope development would be so simple, but the truth is there are no policemen during embryo development. So how do cells know what to do and where to go? This is really a fundamental question. If you think about it, we need to specify and to form cells of the heart in where the future heart will be. We don't want cells of the heart in the brain. So how is everything coordinated? How do embryos coordinate the increase in size, the generation of shape, and the generation of cellular diversity. This is the key question that my lab is trying to address. But now, which kind of techniques we can use, which kind of methodologies, especially if we are talking about human embryo development? How can we actually study this in human embryos? So again, going back to the historical perspective, scientists have tried to study human embryos for really long, and I think really the beginnings of studying human embryo development come with human embryo collections, which were created in many different parts of the world, and especially famous is the human embryo collection of the Carnegie Institution in Washington. In this collection, scientists managed to obtain more than 10,000 human embryos that were developing in vivo, inside the womb. Now, we can leave it for the questions how this was actually possible, how they managed to obtain so many human embryos that were in the womb of the mothers. And these are the historical images from those publications where the morphology of these embryos was reported. So what you see here in number 1 is a 2-cell stage embryo. This is one cell, this is another cell. So this is day 1 of development. Here you see a day 6 human embryo. This is just before the embryo implants into the womb. And then this would be a day 9 human embryo already implanted, so buried inside the uterus. So this is really informative for us because we know how embryos look like when they are developing in vivo. It's like an encyclopedia of human development. But if we want to move forward, we need alternative methodologies. This is just informative. We can look at it, but we cannot do anything with these images. So, of course, a huge revolution and a game changer in the field was the development of human in vitro fertilization. And this is the image from the original publication in 1969, where you can see here, this is the sperm, this is the head, the tail, and that's a human egg. And this is happening, of course, in vitro. This was so transformative that Bob Edwards, the person that developed human IVF, received the Nobel Prize in 2010 for the discovery. And actually, this was done at the University of Cambridge. So IVF has been a revolution. This is clear. It has been a revolution in terms of reproduction. This is Louise Brown. You are seeing here the first IVF baby. She was born more than 40 years ago. She's healthy. She had children of her own. And the estimates are that nowadays more than 10 million babies have been born through IVF. So, of course, IVF revolution for reproduction, but also huge revolution for research. Because now we can look at human embryos developing in the lab. So you are seeing here a zygote is dividing now into 2 cells. Those 2 cells will divide into 4, so on and so forth. You have generation of more cells, increasing cell numbers. Then we have generation of tissue shape. The embryo has to make a hole in the center. And then this structure that you see around, this is called the zona pellucida. It surrounds the embryo, and once the embryo is ready to implant, it's going to come out of that. It's going to break the zona pellucida. You can see here it's trying to come out, and it's ready to implant in the uterus. So in the context of an IVF clinic, this embryo, which would be a day 5 to 6, is now ready to be transferred to the mother so it can continue development inside the womb. So with IVF, now we can look at the first 5 to 6 days of human embryo development. We can see what is happening, we can see what the cells are doing, and we can actually study it. But what happens afterwards? What happens once an embryo implants? This was really a black box, because the moment we put the embryo back to the mother, we don't see anymore what is happening. So this is the next step. How do we take human embryo research to the next level? And I'm going to talk about 2 things. I will tell you about the development of a culture method that I did during my postdoc that allows us to look at human embryos developing in vitro beyond day 7. And then I will tell you a little bit about how can we model human embryo development with stem cells. So can human embryos develop beyond day 7 in a dish? Because when we started this project, we didn't know what was the answer to this question. Maybe they cannot. They actually need the interactions with the uterus to continue developing. So what we did was simply apply all the knowledge we had gained studying mouse embryos, and this is something that I really want to emphasize, how important it is to start working with more tractable species, such as the mouse, gain knowledge, and then apply that to the human, and this is an approach that we use a lot in my lab. So knowing everything we knew about mouse embryo development, we just used the same culture methodology that had been developed for mouse embryos and applied that to human embryos. And the first thing we saw is that there were different cell types present in the embryo. So we have this cellular diversification, cellular specification that I was talking about. This is a reconstruction of an embryo at day 9 of development with different colors. We are labeling different types of cells. So we have in white the embryonic cells. That's the group of cells that will form the entire baby, the entire fetus. And then in blue and green, we have what we call the extraembryonic cells. Those are the cells that will make the organs that support the pregnancy. So, for example, the placenta. So at this stage, day 9, we can see that these different cellular populations are already present. What else can we see? We see that the embryos grow, as would be expected, from day 8 to day 11 as they grow in vitro. And so how big they are, just to give you an idea, by day 11, they would be as small as a grain of sand. So we've talked about different cell types. We've talked about embryo growth. We also have generation of shape, which was the third critical event that we described at the beginning. So different structures appear in these embryos that are cultured in vitro. In red here, you can see what is the formation of the amniotic cavity. This is really the first step of amniotic cavity formation. What is this cavity? This is a cavity that will surround the entire fetus and will protect the fetus. And so when waters break, the liquid that is present inside that cavity is the liquid that comes out. And here you see the yolk sac cavity. This is another organ that supports pregnancy and provides nutrients to the fetus alongside the placenta. So in this embryos culture in vitro, we were able to recapitulate the growth, the cell type specification and diversification, as well as the generation of shape and different structures that we would expect in vivo. And what was remarkable was that they were doing all of this on their own, without the need of a uterus, without interactions with any maternal tissues. So actually this technology is powerful because it allows us to understand what an embryo can achieve on its own. And that's what we call self-organization. Of course, I don't want to say that we don't need the uterus for embryos to develop. That's actually not the message, but it's just to show what the embryos can do on their own. They also have limitations and they have structures that they cannot form, but it's already remarkable that we get such level of complexity when we just grow them in vitro. So now that means that before we could look at human embryos from fertilization to day 6, 7, but now we have expanded the window of opportunity that we have in the lab to actually look at these embryos and study them. So until when can we culture them? Can we go on and on? Actually, there is a really important limit, which is the day 14. This is the ethical and legal limit for the in vitro culture of human embryos. In the UK, it's completely forbidden to grow a human embryo for research past day 14. Is it technically possible? Most likely, with the method that I established during my postdoc, is not. As we can see, the efficiency of development drops dramatically after day 11. But potentially in the future, with the development of new culture methodologies, it may be possible. But of course, we have to again remember there is this current ethical and legal limit, and there are discussions going on in the UK as to whether this limit should stay or we should expand it. So why day 14? I would just mention 2 specific reasons. This is really when embryonic cells start to decide their eventual fate. So this is when they start to take decisions like, do I want to become a cell of the brain, or do I want to become a cell of the liver? And this happens during a process that maybe you heard before talking about it, is gastrulation, really when cells decide their fate. And there is a second reason. Day 14 is the limit for twinning. So an embryo can split into 2 before day 14 and give rise to 2 identical twins. So if we are thinking in terms of personhood, if the embryo still can give rise to 2 different individuals, it was believed that then we could not assign personhood to an embryo until after day 14. You are, of course, welcome to have your own opinion and to think about it, and actually there is a current public discussion as to what this limit should be. Okay, so we've talked about embryo culture. This is great. We can now study embryos past day 5, day 6 until day 11, 12 in vitro, look at them, see what's happening with them. But the way in which we really deep down into what are the mechanisms that control development, which molecules are involved, which genes, which proteins, is usually by eliminating a specific gene or eliminating a specific protein. And as you could imagine, doing that in a human embryo would be extremely difficult. So do we have any other alternatives where we can really study the function of proteins or genes? The alternative would be to use human embryonic stem cells. So I already told you how embryos at these early stages, they have two populations of cells, the embryonic cells that form the entire baby and the extraembryonic that support the pregnancy. So what we can do is we can take those embryonic cells, which I'm showing here in blue, sorry. We can take those embryonic stem cells and culture them in vitro to generate human embryonic stem cells. So human embryonic stem cells, they can make more copies of their own, so that means they are immortal, they grow forever. So once we establish a line of human embryonic stem cells, we can just grow it in the lab, share it with any other scientist in the world. We have human embryonic stem cell lines in the lab that were derived in the 90s, they are still growing. But what is really critical about them is that they can form any cell type of the organism. That's what we call pluripotency. And that reflects their potency in the embryo. In the embryo, they are there to make the entire organism. So it makes sense that if we culture them under the right conditions, we can still generate all the cellular diversity that is present in the organism. And this is really shown with this cartoon, really emphasizing how great, what a great tool they are. If, for example, we need brain cells for regeneration and to treat a patient that has a degenerative condition in the brain, we could potentially get those embryonic stem cells, make brain cells, and use those brain cells for regeneration. So during the past 20 years, people have been trying to generate really pure cultures of these cells that are differentiated to the cell type of interest that then could be used for regeneration and treat patients. But in the past years, there has been a change in the way we think about human embryonic stem cells. So rather than using them to generate specific cell types of interest, since they can make all the cell types of the organism, could we use them to recapitulate the entire embryo? So why not doing the entire embryo instead of just the brain or just the liver? So these are what we call stem cell-based embryo models. Embryonic stem cells are really the starting point, and then by playing with the culture conditions and the kind of compounds we give them and the nutrients we provide and the shapes in which these cells grow, we can mimic different aspects of human embryo development. And not to go into the details, but this is just a collection of pictures where you can see some of these models that recreate certain things. So they can recreate the morphology of a specific developmental stage or the cell type composition of a specific stage. So maybe at this point you are thinking, wait a second, what are you talking about? What does this mean for the future? Reproduction with no eggs, no sperm, no womb? Okay, let's try to digest this step by step. We've talked about these stem cell models of the embryos. So maybe you are wondering, are they really embryos? Are they models? How good they are? But to address this question, maybe we should first try to understand what is an embryo. And this is an extremely difficult question to answer. And just to give you an idea of how difficult it is to answer this question, I'm going to give you the definition that the Human Fertility and Embryology Authority, the HFEA, so this is the body that regulates human embryo research in the UK, the definition that they have. An embryo is a live human embryo. So we haven't learned anything. So how can we define an embryo? Should we define an embryo based on how the embryo is made? Fertilization? Well, no. And I will give you one clear example of why not. Probably you all know Dolly the sheep. And yet Dolly the sheep was not created by fertilization. It was done by cloning. So putting into the definition the way in which an embryo is created wouldn't work. So then what is the alternative? Many people think about potential. To know if an embryo is an embryo, you should have a potential to give rise to an organism, right? And I guess this is the closest we can get to a definition of an embryo. But I personally think it also has limitations because we have embryos in the lab that have a lot of alterations, chromosomal abnormalities, they have defects, We know those embryos have no potential. They would never give rise to a baby. And yet they are embryos. They were created by IVF. So this is just for you to think a little bit about how we can actually define an embryo. But let's stay with the definition of potential. An embryo should have a certain potential to give rise to an organism. So then, what is the potential of these stem cell-based embryo models? Can they give rise to an organism? Well, actually, that experiment is forbidden. The ICCR, the International Stem Cell Research Society, prohibits the transfer of any human embryo model to the uterus of a human or any other animal. So the test that would define whether these stem cell models are embryos cannot be done. So are they embryos? What can we do to address this question? We have alternatives. These are not perfect, but this is what we can do at the moment. We can work with other species. So I've talked about human embryo models. We also have stem cell-based embryo models of mouse embryos. And what we know is that those models have no potential to give rise to a mouse. We are not close at all to making a mouse out of stem cells. We also have monkey models, and again, those have no potential to make a monkey. We can also work with in vitro development. We could take these models and try to grow them in vitro. So we take those human embryo models, can we grow them in vitro and try to generate a structure that would appear later on? Usually they fail. So what does that mean? That based on the current evidence we have, stem cell-based embryo models are not embryos. Maybe in 30 years, I will be here and I'll tell you that's not the case anymore, but as of today, stem cell-based embryo models are not embryos. So for us, they are just an extremely useful research tool because we have embryonic stem cells, we can manipulate them, we can eliminate genes, we can eliminate proteins, and then we can see what are the consequences for development. What happens to those models? What fails? And by doing those experiments, we can understand what is the function of the gene we have eliminated or the protein we have eliminated. And this is how we gain really a deep understanding of how embryos develop. They are also great because it means that we can actually, in the case of mouse, decrease the number of embryos, of real embryos of mice that we need for research because we can use our embryo models. And so to finish this presentation, I will just show you a couple of slides for you to understand the kind of experiments that we are doing in the lab and how we are using these models to understand embryo development. So now this is a mouse. I've been mostly talking about human, but my lab also is doing a lot of mouse embryo research. We start with the mouse. It's a more tractable species. We know much more about them. And then the knowledge we get from mouse can be transferred into humans. And this is the way we work. So this is a mouse embryo at day 6.5. This is when that event that I talked about, gastrulation, takes place. This is when cells start to decide their fate. So now in magenta here, I'm showing you cells that are already starting those decisions. These magenta cells want to become organs such as the liver and the intestine and the pancreas. So those are really the first decisions they are taking. And what we wanted to understand in the lab is how they know they need to appear here in this region and not in another region of the embryo. So again, mouse embryos are also difficult to manipulate genetically. If we want to eliminate a gene, we have to generate a genetically modified mouse. This involves a lot of time, but also a lot of mice that are needed for research. So instead, what we did was to generate a very simple model with embryonic stem cells. These embryonic stem cells grow in 3D, and we have those magenta cells appearing. So now we can actually study how they know where to appear. And so, without going too much into the details, what we found in this study was that as cells start to make more copies of themselves and the tissue starts to grow, they start to be very crowded. There are many cells in the embryo, and so they start to push each other. And by pushing each other, some of the cells are kicked out. As you can see, this one is leaving. And by leaving the tissue in which they were, they start to express the markers of differentiation. So this is really putting together the position of that the cells acquire and the kind of genes they start to express and the final identity. So it's a nice example of how the embryo is coordinating the change in the shape and the generation of a new cell type. So this is about what happens during normal physiological development. Everything goes well. We have changes in tissue shape. We have generation of cellular diversity. But the truth is that, again, going back to human reproduction is very inefficient, and there are lots of errors. And this is shown in what has been called the pregnancy loss iceberg. So it is estimated that from all the conceptions that take place, only 30% of embryos make it to a live birth. And around 70% are lost at some point of development. Most of them preclinically. What does this mean? These embryos are lost before a woman even knows she's pregnant. And that's why it's called the iceberg. We don't really see all these losses. So failure to develop during these earlier stages of development is the major cause of pregnancy loss. Why is that the case? Why is this happening? What we know is that chromosomal abnormalities could be one of the major contributors to pregnancy loss. What are these chromosomal abnormalities? Probably you are all familiar with Down syndrome. Down syndrome is a case where there are 3 chromosomes 21 instead of 2. But there are many other chromosomal abnormalities. The case of Down syndrome is very special because this is one of the very few ones that makes it to a live birth. All the other chromosomal abnormalities would lead to pregnancy loss. And in 50% of early pregnancy losses, we can detect chromosomal abnormalities. And incredibly, in 70 to 80% of in vitro fertilized human embryos, chromosomal abnormalities are detected. This is incredible. That means that at day 5 of development, around 70 to 80% of human embryos have some abnormal cells with an abnormal number of chromosomes. And yet, when the baby is born, less than 1% of babies will have abnormal cells. What is happening in between? So the most plausible explanation would be, well, all those embryos are eliminated. You just told us reproduction is very inefficient. Human reproduction is very inefficient. They are just being lost. And this is why when the babies are born, the cells are not present. But actually, things are much more complicated. So we know that healthy babies can be born from some of those embryos that have abnormal cells. So there have been some clinical trials done where embryos that had abnormal cells were transferred to mothers because those mothers didn't have any normal embryos. So that was the last chance. And from those embryos containing abnormal cells, healthy babies were born without abnormal cells. So what is happening? How does the embryo recognize the abnormal cells? How are the abnormal cells eliminated? This is something that we are trying to investigate in my lab. And so all put together, we are really interested in what happens inside the womb that we cannot really see, how the embryos are being made, how cells take the right decisions, how shape is generated, how embryos grow, how everything is coordinated, and what happens when errors occur. And with this, I'd like to thank everyone in the group. They are a terrific team to work with. And of course, a special thanks to everyone here, all the collaborators, scientific facilities, support services. I see many familiar faces, and I really thank you all for all the support that you give us, because thanks to you, we can do this nice work. And so with this, I would like to take any questions that you may have.
Thank you very much, Marta. So questions? Very quiet today. I'm surprised. Well, I'll start with one. So you said day 14 in the UK. Is it the same everywhere? Is there agreement across?
In some, well, I mean, here is a legal limit. In other countries, it's a recommendation. Other countries don't have a specific limits. For other countries, like Switzerland, is day 7. And in other countries, human embryo research is completely forbidden, like in Germany. So there is huge heterogeneity across the globe.
Marta, you said the magenta cells were going to be going to make a specific organ. Is that what you said?
Yeah, well, it's actually the internal organ, so it would be things like liver, intestine, pancreas, those. But those magenta cells cannot make brain anymore, for example.
Okay, so does it always happen in the same sequence, that those organs are the ones that are made first?
Oh, yeah. Well, I mean, that's a very good question. So during this event that is called gastrulation, you start having those decisions, and some of the first decisions are those magenta cells, that the ones that are taking them. but the other cells are also starting to change. The thing is that in that embryo, I'm only looking at those specific cells. I'm not using the markers of the other cell types, right? So it all depends on when we have the embryo in the lab, what kind of cell type we are actually looking for, if you see what I mean. If I would be looking for other cell types, you would see them in other parts of the embryo, right?
Developing at the same time.
Yes.
Okay.
So with regard to the abnormal cells that can develop into a normal embryo, so do you know anything about that process yet?
Yeah, so there is a lot of controversy in the field of what could be happening to them. One option is that they get corrected, and there are a couple of publications that have shown correction, because, and I can explain this in very simple terms. So in all the cells, for each chromosome, you will have a copy of your mother and a copy of your father, right? So now let's imagine one cell lost the copy of the father. So this cell would be abnormal, it only has one chromosome. And actually having only one chromosome is very detrimental. But now the cell manages to correct because it makes a copy of the maternal. Now you have two maternals. This is something that can be seen in the lab. So we can see that the two copies are maternal. If the two copies are maternal, that means correction. So there are a couple of publications showing examples of correction. I don't think this is really the main way in which these embryos can actually develop further. The other alternative is that those cells are dying. And there is some evidence supporting that's the case in the majority of the embryos that make it, those cells are actually dying.
And can you see, so you said there's some IVF births that have come from like cells with abnormalities. Have those organisms been studied to see if they have any, like, two copies of the maternal?
In those cases, no, it was not a case of correction, but the abnormal cells were not there. The thing is that you know what happens at the beginning and you know what happens at the end, but you cannot see what happens in between because the embryo is developing in the womb. So the only thing you can tell is the cells are not there anymore and there was no correction. So most likely those cells were eliminated. And the other cells that were in the embryo, which were normal, were able to continue developing and compensate. And actually, just to finish this question, that means also that the normal cells that remain, they know now there are less cells in the embryo, and then they can compensate for that and grow more. And we see this very clearly when we do studies with mouse embryos. We can actually separate half of the cells of a mouse embryo. So we have what is called a half embryo. It lacks half of the cells. And that half will sense, oh, there is half missing. And the cells will create more cells. So the final embryo has the normal size. So yeah, embryos really can accommodate lots of changes and continue developing.
You were showing, so how do the cells know that they're going to be, how does that decision start happening of when they're going to be a liver cell or something? And you said it was about crowding and squeezing out. But then also the embryo, it's got different spaces in it. So is it only a particular location where they're squeezed out, those cells become parts of the gut? And if it's somewhere else in the embryo, it's a different kind of cell. Is that part of it?
Yes, and actually, you are absolutely right. That actually, I didn't go into the details, but that process of squeezing and cells leaving can only happen in a very defined region of the embryo, because in all the other regions, there is a barrier for them. It's called the basement membrane, but never mind. It's just a barrier so that the cells, they just cannot, it's like a wall. And in that particular region of the embryo, the wall is broken, so they can actually be living.
You talk about reproductive efficiency, but is that the right way of describing it, really? Because this is more a sort of quality control system. So the whole process is ensuring that what you get at the end is viable and healthy.
You are absolutely right.
If you start to, for instance, if somebody started to interfere with that process to make sure that embryos weren't rejected, you can actually end up with a worse situation.
You're absolutely right, and many people are thinking along those lines. Of course, for humans, it's a huge investment to have a pregnancy and a child, right? So, from an evolutionary point of view, perhaps you want to maximize the chances that embryo fetus is optimal in terms of quality. And so, in a way, that could be seen as a checkpoint in the womb. We just want the best embryos. And there is a theory out there that is quite interesting. So there is a clinical condition called recurrent pregnancy loss. So these are women that get pregnant really quickly, but then they have lots of pregnancy losses. So there is an idea out there saying that perhaps the mechanisms in the uterus that are sensing the quality of the embryo are failing. And so any embryo can implant. And then so you have all those pregnancy losses. So that would be exactly like, you want that, right? You want those mechanisms to select very good embryos. But on the other hand, if we look at mice don't have chromosomal alterations. Less than 1% of embryos would have them. So why in human embryos there are so many? So in that sense, we are inefficient, right?
I can understand,
very good point.
You know, the desire to understand how it works. Because, as you just said, if you've got an overactive quality control department, that's not a good thing. To reset that would be helpful. Could I ask another question? With the 14-day limit, have you got one thing that you would argue for a longer period? One thing that you would want to do, find out? If you had, say...
You mean to extend the limit?
Yeah. What a reason, a pressing reason to...
Yeah, so, I mean, the most pressing reason at the moment is that this process that I've been talking about, gastrulation, when really those decisions are taken, when the beginning of the cell types start to appear, happens after day 14. And we know lots of congenital malformations down the line have their origins at gastrulation. So gastrulation is this really critical point of development. Actually, there is a very famous quote from scientists that said, "Is not birth, marriage, or death the most critical point in your life is gastrulation." So this happens after day 14, and I think is one of the main reasons why the community is trying to extend the limit extra 7 days.
Yes, thank you.
How different are the culture conditions required after implantation?
Compared to pre-implant, is completely different. Completely different, because basically during pre-implantation, the embryo has the nutrients stored in the egg. So they are just using everything that is stored in the egg, but as soon as they need to implant, then they need the nutrients from the outside. So yeah, completely different.
Have there been any major technological advances that have sort of made the culture process easier in the last 20 years since I used to work in a pre-implantation lab, so but I'm just curious to know what's changed.
You mean in the pre-implantation culture or in the post-implantation?
Either, actually.
No, the pre-implantation culture, I think the medias haven't really changed much. They are quite well established, and actually we know, at least for mouse, they work really well, right? Because we can, and actually for humans, you can get pregnancies, but with mouse, it's very efficient. You can take mouse embryos, put them back in female mice, and they give rise to babies. The post-implantation one is more of a work in progress. And I think one of the challenges moving forward is to make the cultures more three-dimensional. So the way it's working at the moment is that the embryo is just going to attach to plastic. So what I like to say to explain it is that the embryo continues developing without implantation. We are not mimicking implantation at all. Implantation is such a complicated process where the embryo talks to the cells in the womb and really invades the womb, right? It's not something we can actually mimic in plastic, where the embryo just will touch the plastic and grow on the plastic. So I think moving forward, more complex culture methodologies where we can include this three-dimensional aspect and the embryo can actually invade will improve those cultures quite a lot.
Brilliant. So if there's no further questions, I'd really like to thank Marta. And our next seminar is on the 24th of March. The Grest.